Hello students, this video will cover all the science process skills that you need to master for your Paper 3 Biology SPM. Now let's begin. We will look at the Paper 3 Biology format before we go into science process skills. The items type is subjective and the number of questions is only 1. The total marks is 15 and the duration for your paper is 45 minutes, including 5 minutes for you to check the apparatus and materials. And the context is from your DSKP, KSSM Form 4 and Form 5 Biology. Now, what are the science process skills that can be asked? Let's check together. The first science process skill that you need to master is observing. Observing will be using your senses of sight, hearing, touch, taste or smell to gather information about objects or phenomena. Now, look at this example. Observe the picture. In test tube A and test tube B, what can you see? Okay, in test tube A, there is an orange-red precipitate form. In test tube B, it is a blue solution. Classifying is the next science process skill that you are going to look into. Under classifying, you are using observation to group objects or phenomena according to their similarities and differences. Now, we look at this example. This question says, classify the following food samples into reducing sugar and non-reducing sugar in table 2. So you have to identify among the food given which are the reducing sugar and which are the non-reducing sugar. Okay, in order to classify this food whether they are reducing sugar or non-reducing sugar, of course you need to identify what type of carbohydrate this food contains. For example, grain contains maltose, milk contains lactose, sugarcane contains sucrose, sugar beet contains sucrose, honey contains fructose, and grapes contain glucose. Okay, as you can remember, all the monosaccharides and disaccharides except for sucrose are reducing sugar. So we can put grains, milk, honey and grapes under reducing sugar and sugarcane and sugar beet under non-reducing sugar. Next is measuring and using numbers. Under measuring and using numbers, you need to make quantitative observations using numbers and tools with standard units to ensure an accurate measurement, and you are able to record in tables systematically and completely with the correct units. Example, look at this experiment. Diagram 1 shows apparatus set up to determine the relationship between the rate of diffusion and different solution concentration. So what are the solution that we are going to use in this experiment? If you look at the diagram, there is an agar strip and the solution is hydrochloric solution, HCl solution. So the agar strip in diagram 1 is prepared by mixing agar powder with sodium hydroxide and phenolphthalein solution will change color to pink in the presence of alkaline solution. The presence of acid, which is in this case is hydrochloric acid, will neutralize the alkali and cause the pink color of phenolphthalein to decolorize. So how are we going to measure and record the data? We are going to use the table. As you can see in the first column, it is the concentration of hydrochloric acid with the unit molarity, capital M in the bracket, and you have 0.2 m, 0.4 m, and 0.6 m. And the responding variable in the second column, which is the time taken for the pink color of agar strip to decolorize, and the unit is second in bracket. The next science process skills is inferring. Inferring means you are able to generate multiple possibilities to explain a complex situation. Inferring also means able to explain the relation and trends between observed variables with measurements used in the investigation. Now we look at the example of the inference question. Look at the table. In the table, you have the first column, the manipulated variable, different concentration of hydrochloric acid, which are 0.2 molar, 0.4 molar, and 0.6 molar. The second column is the observation, that is the responding variable, time taken for the pink color of agar strip to decolorize in second. And the third column is also your responding variable, that is your actual responding variable, rate of diffusion in molarity over second. So to answer the inference question, you take the value from your manipulated variable, but do not take the value from your responding variable. Okay, you have to explain it. Okay, look at the answer. Time taken for the agar strip to turn colorless in 0.2 molar hydrochloric acid is the shortest. Diffusion rate of the molecule is the slowest. You can also use 0.6 molar. If you use 0.6 molar, your answer would be the time taken for agar strip to turn colorless in 0.6 molar hydrochloric acid is the longest. Diffusion rate of the molecule is the fastest. Next science process skill is predicting. Predicting is making forecasts about future events based on observation and previous experiences or collected data. This is an example for predicting questions. Okay, look at the diagram. In the diagram, you have distilled water in the beaker, and in the whisking tubing, you have 15 ml of 30% sucrose solution, and there is a capillary tube inserted into the whisking tubing. So the questions: if distilled water in the beaker is replaced with 60% sucrose solution, predict the level of 30% sucrose solution in the capillary tube of this experiment. Explain your answer. Okay, Is your answer the same as my answer? What is the answer? Okay, the level will decrease or going down. Why? Okay, this is the explanation. P2 is the explanation. 60% of sucrose solution is hypertonic than 30% sucrose solution. Concentration of water molecule is higher in whisking tubing than in the beaker. Or you can also use water potential. Water potential in the beaker is higher than in whisking tubing. This causes water to diffuse out of the whisking tubing by osmosis. Next is communicating. In communicating, you are able to present the experimental findings and observation data in various complex form using graphic symbols such as tables, graphs, diagrams, or models to show relationship between associated patterns. This is an example communicating using table. Okay, so if you look at this one, I already labeled it for you. The first column is the manipulated variable, and the unit is also compulsory, and it is written in a bracket. For the second column, it is the operating responding variable, ORV. That is the time taken for pink color of the agar strip to decolorize. And the third column is also responding variable, but it is the actual responding variable whereby you have to calculate. In this case, it is the rate of diffusion, m, that is molar over second. Again, it is written in a bracket. Now, look at the data. Okay, for the first column, you have 0.2, 0.4, 0.6. all with one decimal point. And the second column, you have 20, 15, and 11. That is the time taken in second. And for the rate of diffusion, you have 0.01, 0.03, 0.05. Make sure all your decimal point is consistent. Okay. This is how you communicate your data using graph. The general method of drawing a graph. Okay, the responding variable is represented by the vertical axis, that is your y-axis, and manipulated variable is represented by the horizontal axis, that is the x-axis. The scaling of the axis must be uniform, and all the points must be marked using symbol X, and it must be clear. The title of the graph would be Graph of your responding variable versus manipulated variable. For example, in this case, volume of oxygen release versus temperature. Okay. For this kind of graph, that is line graph, okay, you only connect the points. Do not connect below or beyond the points. In other words, do not extrapolate. The hypothesis for line graph is the higher the temperature, the higher the volume of oxygen release until the temperature reaches 40 degrees Celsius. This is another example of communicating using graph, but this one is a bar chart. Just now it was a line graph. This one is a bar chart, but the axis remain the same. The manipulated variable is at the x-axis and the responding variable is at the y-axis. For the bar chart, the hypothesis is a little bit different. For example, like this one, almonds has the highest temperature change, followed by cashews, pecan and hazelnut. For the bar chart, make sure there is space between each of the bar. This is another way how you communicate using a diagram. If you look at this diagram, all the apparatus use symbol. Okay, never draw your apparatus using a 3D diagram. Okay. Okay, this is the end of Science Process Skill Part 1. Please continue to the next video for the SPS Part 2.